Hello guys, we are from Team Kulfi, which consists of me, Daniel, Ayemann, Talent and Longyu. So for today we'll be discussing and talking about the technique known as transcriptomics.
Hello everyone, Assalamu alaikum. My name is Ayemann and I will be telling you about transcriptomics for my SIO 2005 presentation on the aforementioned topic. What is transcriptomics? Now, transcriptomics is a study of the transcriptome. Transcriptome, as defined by naturenews.com, is the complete set of RNA transcripts that are produced by the genome under specific circumstances or in a specific cell using high throughput methods such as microarray analysis. Comparison of transcriptomes allows for the identification of genes that are differentially expressed in distinct cell populations or in response to different treatments. So this is essentially the study of mRNA transcripts produced by the translation process in cells on the ribosomes to better understand how genes work or to identify genes. Now, the history of transcriptome is very brief. The first example of such studies date back to the 1970s, where libraries of silk moth mRNA transcripts were organized and used to produce complementary DNA, cDNA for storage using reverse transcriptase. Sanger method was used to generate sequence random transcripts producing express sequence tags or ESTs in the late 1980s. Now, the word transcriptome was originally used in the 1990s. And in 1995, one of the earliest sequencing-based transcriptomic methods was developed. This was referred to as SAGE, serial analysis of gene expression. Now, the more modern techniques of microarrays and RNA-seq were invented in the late 90s to early 2000s. Now, this about wraps up our history of the transcriptome. Coming up next are our citations for this section, and I will now pass the torch to one of my dear class fellows to continue the presentation. Thank you very much.
Okay, my name is Daniel and today I will discuss about the working principle of transcriptomics. So basically there were two major techniques that were developed in transcriptomics. The first is microarrays and the other one is RNA sequencing. And the main difference between these two available techniques is that they serve a different purpose and both of them have their own specific function. So for microarrays, when microarrays is a technique that we use when we are more interested in the number and the quantity of a specific or targeted mRNA. And this technique actually depends heavily on the usage of complementary probe that will tell you about the number and also the quantity of the mRNA. So if we are more interested to quantitate the specific mRNA sequence, so microarrays is the best method to use. And the second one is about the RNA sequencing. RNA sequencing allows us to sequence the complementary DNA which are produced through a process known as reverse transcription of mRNA. So this method tells us about the sequence of the gene which is responsible in producing the mRNA, and also it tells us about the location of the gene within the genome. So if we are more interested about the sequence of the gene responsible in producing the mRNA, RNA sequencing should be the method to be used. Okay, now let's discuss further about the principle of microarrays. Microarrays can be further divided into three compartments: in vivo, in vitro, and in silico. I'm quite sure that most of you know what are in vivo and in vitro, but what is in silico? So basically in silico is a step of analyzing that is done through a computer. So the first step of microarrays occur within the organism itself, where the DNA will be transcribed and will be spliced to produce a mature mRNA that consists only exons. Next, the mature mRNA will be extracted from the organism, and it will undergo a process known as reverse transcription to produce a double-stranded complementary DNA. Then, the long double-stranded complementary DNA will be further fragmented into shorter fragments where each individual fragments will be labeled with a fluorescent labeling. And then, the labeled shorter fragments will bind to complementary probes that were prepared earlier. And the last type of microarrays is the analyzing of the abundance of each complementary DNA. And the abundance of the complementary DNA depends on the fluorescent intensity of the shorter fragments. Now let's discuss about RNA sequencing. Same as microarrays, it also happens in three compartments: in vivo, in vitro, and in silico. The first one occurs in the organism itself, where the DNA will be transcribed and it will be spliced to produce a mature mRNA. Once the mRNA is extracted, it will be fragmented first to produce a smaller RNA fragments, and then each RNA fragments will undergo a reverse transcription to produce multiple smaller fragments of double-stranded complementary DNA. Then each shorter fragments of double-stranded complementary DNA will be sequenced through a technique which is known as high-throughput sequencing. Once the sequencing is done, the shorter fragments of complementary DNA will be aligned with a reference genome to locate and to determine the specific position of each complementary DNA.
Alright, now let's talk about the known research use. And as you can see here, I have selected the 5 most common applications, which I will be explaining further in the next slides. Firstly, the diagnostics and disease profiling, which has been useful in biomedical research. RNA sequencing have allowed scientists to study transcriptional start sites, in which they have discovered alternative promoter and alternative splicing, which is a mechanism of modification to create diversity and flexibility in the regulation of gene expression. And in studies, they have found that these genes are regulatory elements, which are the driving force of gene expression. And major cause of human disease are actually caused by defects in these elements. Thus, by identifying these regulatory elements, scientists can interpret disease causal variants from single nucleotide polymorphism, allele-specific expression, and gene fusions. And as you can see here, scientists have used these transcriptomic tools to identify disease of tuberculosis. Secondly, the human and pathogen transcriptomes. Humans are prone to infection by opportunistic pathogen due to emergence of novel pathogenic species and lack of effective diagnostic tools. Also, pathogens are phylogenetically diverse, and their virulence mechanism can differ widely across species, and as a result, scientists could not figure out molecular basis of virulence in pathogens and their interaction with human hosts. Thankfully, with RNA sequencing, scientists can understand the molecular mechanism of virulence and host defense. This is achieved by monitoring the transcriptional profile of both human and pathogen. And from the research, we can say that transcriptome sequencing have been used by scientists to understand the interaction between human and pathogen. Next, it is also widely being used in plant breeding, in which this technique have been used to improve the quality traits of transgenic crop by improving the plant response to a particular biotic or abiotic stress. And they are able to understand the plasticity of gene expression response to external stimulus. And from the research paper here, this plant is transformed with the coding sequence of a gene for 3-halose-6-phosphate, which is a sugar known to play a role in drought resistance. And according to the result, when this enzyme is expressed in plant, the transgenic have larger leaves, altered stem growth, and improved response to stress. Moving on, transcriptomic technique have also been useful for gene function annotation. So DNA annotation is the process of adding information such as genomic position, regulatory sequences, and gene names to previously unknown sequence. And from the research paper, RNA sequence has been used to determine the function of unknown genome in commercially important organism, which is the cucumber. And last but not least, transcriptome is also being used in stem cell research. Stem cells are self-renewal and pluripotent. They are known well to cure some refractory diseases such as stroke, myocardial infarction, and diabetes. However, according to studies, stem cells are heterogeneous and consist of many cell subpopulations. In addition, gene expression in stem cell is highly diverse between cells, even within a single tissue, and as a result, it has been difficult to make precise identification and analysis of its biological properties. But with the development of single-cell RNA sequencing, it has become a breakthrough in stem cell research. This is because they can now identify novel cell types and explore cell markers easily. And from the research, the technology of single-cell RNA sequencing is being used to conduct research on embryonic stem cells. And that's all. Thank you.
Okay, I'm Daniel. I'm back with advantages and the disadvantages of transcriptomics. So I'll be discussing this based on the principles that I've mentioned earlier. So now let's take a look at the microarrays. So some of the advantages, notable advantages of microarrays is that this technique allows us to produce a large amount of data at once. And also the operational cost of microarrays compared to RNA sequencing is significantly lower. So we can save a huge amount of budget if we use this technique. But the downside of it is that it is much lower in sensitivity, specificity, and also the resolution. And also since this technique depends heavily on the usage of probe, it is compulsory for us to know about the sequence of our desired mRNA because we need to synthesize a probe that is complementary to our desired mRNA. So therefore, it can be only applied on known mRNA or known gene. Okay, now let's talk about the RNA sequencing. Some of the benefits of using this technique is that it can detect various genes as it covers large scale of genome transcript. And this technique also allows us to study both known and unknown genes. This is because it doesn't really depend on the usage of probe. So if there's any chance of us of finding any unknown or any undiscoverable genes, it is definitely through this method. And since we don't need any probe in this technique, so it doesn't really matter if we know the sequence of a targeted mRNA prior of using this method. And this method has a pretty higher sensitivity and specificity compared to microarrays. But as usually, everything has a downside, everything has a dark side. So some of the disadvantages of using this technique is the sequencing platform. So what is sequencing platform? Sequencing platform is actually a platform that is used to sequence the cDNA. The problem is that there are too many companies that are producing or that are preparing this sequencing platform, and each sequencing platform has their own requirements and are highly specific. So any wrong choice that we made will not produce a great result. The next one is about the cost versus the read coverage. If we want to use a higher read coverage, the cost will also increases. And the last one is about the inefficiency of the fragmentation of the RNA. In the step earlier, I mentioned about the process of RNA fragmentation, and the problem with the step is that this step is highly inefficient. So therefore, we will not be able to get our desired fragments frequently. So yeah.
I'm going to talk about limitations of transcriptomics. As all we know, transcriptomics has emerged as a powerful approach for exploring physiological response to the environment. However, like any other experimental approach, transcriptomics has its limitations. An initial promise was that unveiled screens of the transcriptome would serve to isolate novel or unforeseen genes with large consequences on fitness under different environmental conditions, the so-called gene that matter. In 2005, Fader and Wassel conclude that this specific promise has gone largely unfulfilled, offering a pointed description of the major issues facing the use of transcriptomics in finding the genes that matter for environmental adaptation. Their critique focus on three major issues. The first thing is genes with large impacts on fitness are rare and therefore unlikely to be identified with transcriptomics. The second thing is the relationship between gene expression and the fitness is unreliable. Finally, the third thing is fitness is determined primarily determined by proteins, and the mRNA abundance is a proxy for protein abundance. The goal of the review is to re-evaluate the validity of these statements in the context of new experimental evidence, and also to identify new limitations that may have emerged as a consequence of the increasing popularity of transcriptomics in studies of the environment. In general, data collected over the last decade indicate that these criticisms remain valid today and that new issues may have also arisen. While its information does not discredit the widespread and continued use of transcriptomics within environmental physiology, it does illustrate the types of research questions that transcriptomics experiments are best suited to address. A wider understanding of potential limitation of transcriptomics is especially relevant given that technological advances have made transcriptomics increasingly accessible, and more researchers are likely contemplating integrating this technology into their research programs. Information summarized here may also stimulate research aimed at addressing uncertainties about the use of transcriptomics. A major reason for the starting power of transcriptomics is that the scientific community has often responded to criticisms with targeted research that improved how transcriptomics data are acquired and analyzed.
So, in conclusion, transcriptomics is a method to study the transcriptome of an organism. Two major techniques that were utilized in this field are microarrays and RNA sequencing. Even though both techniques have their own sets of advantages and disadvantages, their large contribution to this field should be appreciated. Here's our list of references. That is all from Team Kulfi. Thank you.